The final part of the analysis is pathway analysis. So to do this, first you need to install cluster filer, a profiler, a path view and summarized experiment as 3 different libraries. And when you looked at the data to start with, all of the genes were named by ensemble IDs. So what you need to do is now convert that onto entree IDs, which is what the next part of the code does. So it filters through that list of differentially expressed genes and finds the new names. So if I look at differentially expressed genes, you can see here's the ensemble gene name, the log fold change, the likelihood ratio, the p-values, the false discovery rate. Best to order them by false discovery rate. So that changes the order, so the actual top gene is SSTR1. So that's possibly worth looking at in Google Scholar. So SSTR1 and breast cancer. So somatostatin receptors in primary human breast cancer, quantitative analysis of mRNA subtypes. So there is some relationship between it. Right, let's go back to here. So let's continue and convert those names here into entree IDs by searching through the database. So this is lots of code to extract the exact columns that you want to do. Extract the row names and then check it against a dictionary and reassign the new names. And then at the end, what I want to do is output them into a genelist.csv. So this is the CSV file that I'm going to have which contains the entree ID. So here it is. If I open that file, I'll see a list of IDs that look like this. So the entree IDs are numerical. Now, there's only 390 of them have been identified within the entree ID from the ensemble IDs. And that happens because different databases have different definitions about what sort of data they're going to have in it. So for example, this might not include any of the non-coding RNAs, which I don't want for this particular pathway analysis anyway. So I'm going to do the pathway analysis in a tool called David. So if I go to David and bioinformatics through Google, they can find this David functional annotation tool. So what I want to do is start an analysis, and I want to upload a list of genes. So I'm going to paste in those genes names that I copied. It's an example of entree IDs, and this is a gene list. So I can submit this list to David, and it says all 390 of them have been identified from Homo sapiens, which is good because it's human data. Then to actually do the analysis, I am going to do functional annotation tool. So what this is done is like the genome set enrichment analysis. It's gone to the databases and it's searched against the gene ontology, which we've done already, we don't need. But the extra ones that I want to look at are pathways. Now it's done it against 3 databases, BBID, BioCarta and KEGG. I want the KEGG data. So I click on the KEGG chart. Now here I want to look at which are the pathways which are statistically significant. Now, after correction for multiple testing, the only one that is significant is neuroactive ligand receptor interaction. If I was going to ignore the multiple testing and just to do the p-value without multiple testing correction, Then there's a few more pathways that are included, including taste transduction, which is interesting, metabolism of xenobiotics by cytochrome P450, and chemical carcinogenesis. Apart from that's not quite significant, so probably you'd have to stick here even if you went for the p-values that are there. But the key thing that I wanted is to show what happens. You click on the pathway and it gives you this diagram with lots of flashing things. So what this is telling you is that these particular genes are changed within this pathway. So there's something that's changed in KISS peptide recognition, neuropeptide S, calcitonin, GABA receptors. Now, while this is interesting, and I can stop the flashing by pressing the top flashing things, and I could save this image, what I'd want to see is the levels of change in gene expression coloured onto this chart. Now this chart happens to be the 04080 pathway within K. So what I can then do is go back to my script, and you can see that I've already found that 0480 is the important pathway, but you can add as many pathways to do the analysis for as you want to. So I'll just run this one to start with. So what you do in this is when you run, it goes to the database, grabs the pathway, and then it combines the data that it's done from this analysis, the gene expression data that's within R, with that pathway, and it creates a new file. So the new file is going to be named after the pathway, so it's this particular thing. And you've got two files here. So one of them is the original pathway, which is all in green, nothing interesting there. And the second one is a version of it which is coloured by whether it's up or down regulated. So we're comparing stage 1 to stage 4. So stage 1 is group 1, stage 4 is group 2. So these things in red go up between stage 1 and stage 4. The things that are in green go down between stage 1 and stage 4. So there's less CHRN and GRI, but there's more GABA, CALCIA, NPS, SSTR, GRM and NPR. So let's get the David analysis and just demonstrate another pathway. Let's look at the motor proteins, because that's actually slightly interesting, because metastasis is going to involve motor movement. There's DNA, dynein and myosin are affected, as well as microtubules and kinesin. This is going to involve the cell being able to move. So this is a nice pathway to draw a diagram of, and it's 04814. So I can go back to my R code. I can copy that block of code. I can change this to 04814 in both the top and the name, and I can run this block, which will now get the 04814 pathway and do the same process as it did before. So now if I look at the 04814, you can see that dynein is upregulated in the metastasis. The other ones are in grey, which means they have been changed, but it's not sufficient to say that there's a strong positive or negative change. You can see that they're probably going to be upregulated, but not full. It's not determined completely. So this is this part of this loopy movement to take cargo around.